Okay, so welcome to Proteins Part 2. This is a supplement or the next lesson to Proteins Part 1, just because we couldn't fit it all into one meeting, and Barry can pause and take in this lesson. Okay, so for the learning points for this lecture, we will continue with the types of proteins based on their solubility and physical properties, and then types of proteins based on molecular length and shape. We will also briefly discuss the physical and chemical properties of proteins, and then talk about denaturation of your proteins, and then an application to your medtech life immunoglobulins. So first up, let's look at the solubility and physical properties of the proteins and the different types based on these properties. So first off, we have the easiest ones, the simple proteins, and they're composed only of amino acids. So if a protein is only made up of amino acids completely, it is called a simple protein. Examples are your globulin, which you will find in the immune system, for example, in your immunoglobulins, but you also have other types of globulins. Okay, that's just an example. Your immunoglobulins. We also have albumin, which is very, very important in maintaining oncotic pressure in your blood vessels. So albumin is actually the most abundant circulating protein found in your plasma, in the blood. And it is again responsible for oncotic pressure. So what is oncotic pressure? See, oncotic pressure, it is the pressure that keeps your blood within the blood vessels. It keeps the fluid within blood vessels because remember, diba, in the concentration gradient, water will be retained in the area where there's a lot more solutes. The albumin is the main solute that keeps the water inside of your blood vessels. Okay, and then hydrostatic pressure is the pressure that pushes fluid out. So, ayon, albumin is the one that will keep the water inside your blood vessels, and that's called oncotic pressure. Next, we have the conjugated proteins. In comparison, the conjugated proteins incorporate one or more non-amino acid units in their structure. So, for example, we have apoproteins. May portion siya. You have your apoprotein and the prosthetic group. The apoprotein is the mismong amino acid group. So, for example, if apoprotein lang, talaga walang prosthetic group, that's just a simple protein. But for conjugated proteins, you will have the prosthetic group, the non-amino acid group. Examples of these conjugated proteins are your glycoproteins that you will see in your cell membrane and glycolipids as well. So these are examples of your conjugated proteins. So hemoproteins will have your amino acid apoprotein group, and the prosthetic group is the heme unit. Lipoproteins, the prosthetic group is lipid. So for example, yung low density lipoprotein, high density lipoprotein or HDL. These ones you will see, diba, in the lipid profile, kasama ng total cholesterol. We also have glycoproteins, as I said, that you will find on the cell membrane. The prosthetic group is a carbohydrate kasi glyco. So prosthetic group, carbohydrate. Examples are mucin, interferon, which is also in the um, immune system, gamma globulins as well. Phosphoproteins, phospho, so phosphate group, nucleoproteins, nucleo, nucleic acids, and metalloproteins, metal, metal ion. Okay, so please take note of this entire table. Lastly, we have your derived proteins. The derived proteins, in comparison to the simple and the conjugated, you obtain them from the partial or complete hydrolysis of those simple or conjugated proteins by physical or chemical means. So hydrolysis can be by acids, alkalis, or enzymes. So for example, we have your proteoses, peptones, small peptides, fibrin, metaproteins, and coagulated proteins. We will talk about these coagulated proteins and the derived proteins on how we get them later on in the denaturation part of this lecture. So you can just pause anytime if you need to, but let's move on to the type by molecular length and shape. First off, we have our fibrous proteins. Fibrous proteins, by as the name suggests, they are fibers. So they are elongated, they are tough, they're insoluble, and they're composed again of fibers and sheets. The axial ratio of your fibrous proteins, or the length versus width, is greater than 10, which means that it is longer more than it is wide. Okay, it's longer more than it is wide. Examples are collagen and keratin fibers. Isipin nyo na lang kasi nasa hair siya. So, collagen and keratin, they're in your hair, they're fibrous proteins. Myosin as well that you find in your muscles, and fibrin, which you find in blood clots, are also fibrous proteins. So again, longer than they are wide, and they are insoluble. Next, we have globular proteins. So globe, circular, spherical. These ones naman in comparison, they are water soluble. They're easier to dissolve in water and they can dissolve in water. The axial ratio is less than 10. Therefore, it is wider than it is long. Okay, again, diba, si fibrous, longer than it is wide. Globular, it is wider than it is long. Examples are myoglobin and hemoglobin, which carry or store oxygen in your blood and in your muscle. So, sa muscle, si myoglobin, sa blood, si hemoglobin. And lastly, we have our membrane proteins, which, as you would probably assume, you can see in the membrane because they are membrane proteins. The examples are ion channels and receptors. So remember our lecture on transports. So yung ion channels, like your co-transporters, mga pumps, membrane proteins yon. This is just a summary table that will give you a comparison of fibrous proteins versus globular proteins. So, diba? Fibrous proteins, long and narrow, globular are round kasi globe. Fibrous proteins, because of their nature, na they're long, they're insoluble, they're strong, they are usually for structure, while globular proteins are more functional for transport, as catalysts, as storage, and then, ayon, fibrous proteins, insoluble, globular or soluble. Sequences of amino acids in fibrous proteins, usually they're repetitive. Kaya their 3D structure is more elongated kasi repetitive lang naman. While globular, usually irregular. So magfo-fold in siya on itself. Fibrous proteins are less sensitive as well to changes in heat and pH. That's why they're very good as structural support in comparison to globular proteins, which are more sensitive. Because imagine if our fibrous proteins that are there for structure are very sensitive to heat and pH, edi magbe-break down yung body natin easily because of that. Okay, so again, examples, collagen and keratin. So imagine, konti lang na heat, masisira na si hair mo. Okay, so you don't want that. That's why we have fibrous proteins in our hair and our skin, while globular proteins are hemoglobin, insulin, and catalase. Those are just examples. So again, these are more examples of our fibrous and globular proteins as well as their functions. Let's move on to the physical and chemical properties of our proteins. In general, proteins are tasteless and colorless, but some are colored and appear in crystalline form. Yung natitaste nyo in the meat, diba? When you eat beef, for example, you want marbling in the beef because the marbling is made of the fat, and fat is the one that has flavor. So fat equals flavor. Proteins themselves don't really have a taste. So, mas masarap yung beef pag mas maraming marbling kasi mas marami siyang fat. Okay, but moderation sa mga steak, moderation. And in general, they are also insoluble in fat solvents, and depending on the amino acids that they have, diba, we have polar, non-polar amino acids, charged and neutral amino acids. Syempre, their solubilities in other solvents would also vary depending on what type of amino acids they are mostly composed of. Most will also have high molecular weight kasi dugtong-dugtong siya na mga amino acids. And as we discussed in general, um, about 50 amino acids yung um, average na length of a protein. So they would have high molecular weight. Most also form non-diffusible colloid solutions of the emulsoid type. So that just means proteins usually exist in liquid and will also be dissolved in another liquid. So those are your colloid solutions of the emulsoid type, mostly. They are also generally amphoteric. Amphoteric just means that they can be both an acid and a base, which we will discuss after this slide. Most are also labile and are readily modified in solution when subject to different physical changes like the pH, UV, heat, that's heat not here, and organic solvents. And because they are labile, they are very reactive and highly specific. So yan na. Amphoteric again means that it can act as an acid or a base depending on the pH of the medium. Why is that? That's because we have a basic amine group and an acidic carboxyl group. Okay. This one is the Zwitter ion form, which we'll also discuss later. So in acidic solutions, your amino acids, which are in your proteins, they will act like a base. Again, acidic solution, they will act like a base. While in alkaline solution, they will act like an acid. So in neutral solution, this carboxyl group will lose a proton and become negative. So this one becomes this negative, while the basic group here, the amino group, will accept protons and become positive. Okay, so that's why it's amphoteric, because it can exist and act as a base or an acid depending on the pH of the medium. Again, when you are in neutral solution, you will have this Zwitter ion form, which has both a positive group and a negative group. So ano ba yung Zwitter ion that I'm talking about? Zwitter ion is just a German term for double ion because it has both a positive and negative charge. And because they have both a positive and negative charge, they will cancel each other out and walang net charge yung magiging molecule mo. The structure of these Zwitter ions change when the pH of the solution changes. If you are in an acidic solution, it will accept protons and be more positive. In a basic solution, it will lose protons and become more negative. Okay, so what does that look like? So this is your Zwitter ion. Diba sabi ko, Zwitter ion, no net charge. It has both a positive and a negative na group. Kaya no net charge. They cancel each other out. When it is in an acidic solution, like pag marami kang hydronium ions, like this one, you will have a positively charged ion because the hydrogen here or the proton here will transfer to this negatively charged carboxyl group. So, ito na siya. Magiging full carboxyl group na siya. And because nawala si negative charge, magiging positively charged yung ion mo. In a basic naman na solution, this Zwitter ion will donate this hydrogen from here to the hydroxyl ion. This one, because this hydroxyl group is very, very basic. So, magiging ganito siya. Magiging amine group na siya na wala si positive charge. Therefore, naiwan na lang si negative charged. It is now a negatively charged ion. Again, take note, when a Zwitter ion is in an acidic solution, such as in solutions with high hydronium ions, they will accept hydrogen or protons. So hydrogen and protons, they're the same in this case, and they will become more positive because they accepted a proton. On the other hand, if a Zwitter ion is in a basic solution, such as those with a lot of your hydroxyl ions or molecules, they will lose their proton or the hydrogen here and become negatively charged. Okay, take note of that. So this is just a reiteration of what I said. Yan. In an acidic solution, your zwitter ion will be positively charged, and in a basic solution, your zwitter ion will be negatively charged. Let that sink in. Pause if you need to. Now let's talk about the isoelectric point. The isoelectric point is the pH at which an amino acid has no net charge. So at isoelectric point, the Zwitter ionic form of the amino acid will exist because the isoelectric point is when there is an equal number of positive and negative charges in the molecule. Diba? This is the very definition of a Zwitter ion. And your different amino acids will have different isoelectric points. So this is the pH. So at pH 6.01, alanine will be in its Zwitter ionic form kasi yan yung isoelectric point niya. Arginine at pH 10.76 will be in its Zwitter ionic form because at that point, magiging equal yung positive and negative charges niya. Okay, that's basically what it's saying. All of these, again, are pH at which the amino acid is in its Zwitter ionic form. Now, how do we determine the isoelectric point? Bakit nalaman natin na they have different isoelectric points? We can determine the isoelectric point of an amino acid or basically any molecule by doing electrophoresis. Electrophoresis is separating charged molecules based on their migration towards charged electrodes with a propagated electric field. So if you guys have any background on electronics, electric, or kahit physics lang, basic physics, this one will act like a circuit. The buffer is the solution which provides a medium for your current to pass through. The anode is your positively charged electrode, while the cathode is your negatively charged electrode. The anode, because it is positively charged, will attract the opposite. Kasi 'di ba, opposites will attract. So the anode will attract anions or the negatively charged ions. The more negative a molecule is, the closer it will be to the anode. While your positively charged molecules or your cations will be more attracted to the cathode, the negatively charged electrode. Huwag ma-confuse ha. The electrodes charges are opposite to what ion they will attract. Okay, so cathode negatively charged, anode positively charged. The isoelectric point, again, this is the point at which the amino acids will migrate equally to both directions. Kasi 'di ba, again, the isoelectric point is when there is no net charge. So, yung amino acid mo dapat pag dumating siya sa isoelectric point mo na pH, okay, itong buffer, pwede mo i-change yung pH. Pag yung pH ng buffer na to will be in a pH at which yun pala yung isoelectric point ng amino acid, the amino acid will basically not move. Kasi equal lang yung kanyang migration towards the cathode and towards the anode. Wala na siyang charge. So for example, we have here a moistened paper na meron tayong lysine, phenylalanine, and glutamate. Okay. Pag ito, tinurn on natin si electrophoresis machine, we have our positive electrode, your anode, and the negative electrode, your cathode. Once current passes through this, magkakaroon ka ng electric field. Now, si lysine, magmo-move siya towards the negative electrode. Because what is lysine again? Diba, it is a basic amino acid. So the basic amino acid is positively charged. Therefore, it will be more attracted to the negative electrode. Phenylalanine is neutral, so it will have no movement, while your glutamate is what? It's a what? amino acid that is acidic. And acidic amino acids are negatively charged. Therefore, glutamate will be migrating towards your positive electrode or the anode. Okay? So, repeat nyo lang to na part ng video if you still don't get it. This is why I told you guys, you should always remember the types of amino acids. Remember nyo na lang yung song that I gave you. That will help you. Okay? Now let's move on to the denaturation of proteins. So the denaturation of proteins is the partial or complete disorganization of the structure of a protein up to the secondary structure lang. This will not affect the primary structure. Kasi yung primary structure, it's held together, diba? By what type of bonds? Peptide bonds. So those peptide bonds will not be touched by denaturation. Kaya niya lang until secondary structure. Tapos gaganyanin niya lang. Basically, it will unfurl your protein. Different denaturant factors are temperature, pH, organic solvents, heavy metals, and mechanical stress. There are times when you can actually renature a protein so you can put this back together, but most times, pag denatured na siya, very mahirap to put it back together. First, let's talk about temperature. As you would know, dapat from your physics, temperature is directly proportional to entropy. What is entropy? Entropy is the disorganization in a system. So if you increase your temperature, entropy will also increase. Okay? So, shake up yung inyong structure ng protein, and basically because there's increase in entropy due to the increase in temperature, magkakagulo-gulo yung structure ng proteins ninyo, resulting later on into coagulation. This is unfolding and then entanglement of your proteins. Si denaturation, siya lang yung unfolding. But the entanglement after the unfolding is the coagulation. And coagulation usually is above 50 degrees Celsius for most proteins, nasa mga 56 to 60. And coagulation basically just means stiffens or hardens. And this is what happens to most of our food when we cook it. A very good example is the egg. Diba when we start cooking an egg, the egg is still, yung white part ng egg especially, it's very clear. You can still see through it. But after you cook the egg, temperature increased, entropy increased, na-shake up yung structure ng proteins, na-unfold siya, tas nag-entangle with each other, kaya nag-stiffen and nag-harden si egg white. So, ito yung sinabi kong difference ng denaturation, so nag-unfurl siya, but then with increased heat pa talaga, nag-coagulate si protein, nagkagulo-gulo na, nag-entangle yung proteins mo. It no longer went back to its original helical structure. Gulo-gulo lang siya. Next, we have our pH. pH changes the stability of the structure that leads to unfolding. Isipin nyo na lang, kasi 'di ba dun sa Zwitter ionic form ng inyong amino acid, nagche-change siya 'di ba? Pag nasa acidic na solution, nag-accept ng protons, magiging more positive siya, while in a basic solution, it will lose the protons and become more negative. That's why if the pH of the solution is more basic, basically when it's greater than the isoelectric point, the net charge of the protein will be more negative, while in a more acidic solution that's less than the isoelectric point, it will have a net positive charge. And because of those changes in the charges of your amino acids in the protein, madi-disrupt yung structure niya. Kasi, if you remember, sorry, the structure of your protein is also, the 3D structure especially, is very dependent as well on the charges, on the repelling of the like charges and the attraction of the opposite charges ng different amino acids nyo in the protein. So, syempre, pag change yung mga, change yung charge ng individual amino acids, syempre, change din yung 3D structure ng protein nyo. Next, we have organic solvents, which will disrupt hydrogen bonds. And if you remember, hydrogen bonds are also one of the key factors that hold together your secondary structure of the protein. So it holds together the alpha helices and the beta sheets. Examples of your organic solvents are alcohols. Kaya we can use alcohols to decontaminate spaces because it will break down proteins ng inyong bacteria and other na mga proteins na naiwan dyan na dirty. Okay? Kaya pwede yung alcohol. Detergents can also be used to clean because they also disrupt hydrophobic interactions. They are amphipathic. What does amphipathic mean? Amphipathic means that it has a hydrophobic and a hydrophilic region. So, kaya din nagagamit si detergents pang linis kasi nakaka-disrupt siya ng hydrogen bonds and hydrophobic interactions. So, madidisrupt yung proteins na nakakapangit. Okay, so ano bang ginagawa ni detergent? See detergent, an example, sorry for the pixelated image. An example of a detergent that we will use a lot in biochemistry and in molecular biology, we have sodium dodecyl sulfate. Sodium dodecyl sulfate will interact with the hydrogen bonding dito ng inyong proteins. And then, ide-denature niya like this. So, didikit-dikit siya along the structure of your protein such that the protein can no longer assume its original structure. Next, we have your heavy metals. And as we know naman siguro, all heavy metals have a lot of very bad effects to the body. Kaya nga 'di ba wala na tayong mga mercury na thermometers and lead pencils are also not good. We use graphite pencils now, not lead pencils anymore. So heavy metals interfere with salt bridges formed between your R groups and form bonds with negatively charged side chains. So, ano ba yung mga negatively charged side chains? Diba yung mga acidic, yung D and Q. So, yan yung negatively charged side chains. Examples of heavy metals, again, are mercury and lead. Lastly, mechanical stress. So, nasistress din si proteins and it will cause them to break down just like humans. So, physically disrupting the interactions of a protein will take apart its structure. A very good example that we see in everyday life, siguro if you do this or if you've ever done it, is whipping egg whites or even whipping cream. When you beat egg whites enough na na-aerate mo siya and you really put in the work, pwedeng nilagay mo siya in a mixer or you did it by hand, you will eventually end up with this either soft peaks or stiff peaks. The soft peaks that you end up with when you beat egg whites means that you only partially denatured your protein. While if you have stiff peaks, yung magsistay in place talaga siya like this one, that means that you have completely denatured the protein. Okay, so that's an example of mechanical stress denaturing the proteins. This here is just a summary of all that we've already talked about. We didn't discuss much radiation, but and they will also disrupt hydrogen bonds, and ultraviolet radiation will operate similarly to heat. So just take a look at this table and be familiar with it. This is also another table that I would like for you guys to be familiar with. It's similar to the previous table lang, just more examples. Yan yung sabi ko sa agitation, you have whipped cream, meringue made from egg whites, etc. We are now at the last part of this part 2 proteins lecture, the immunoglobulins. And immunoglobulins are very, very relevant to medtechs like yourself, and especially if magme-med din kayo after. So immuno from the immune system, and globulins because these are globulin proteins. Are globulin protein simple or compound? Diba they are simple proteins. So antibodies will look like this. Antibodies are produced by plasma cells, and plasma cells are your mature B cells. So the plasma cells will produce these highly specific antibodies that have memory. It will recognize self from non-self. So, siya yung parang magde-detect. Para siyang guard ng body na magde-detect siya na, hala, invader to. This is not a body cell. This is a bacterial cell. So maybe we should attack it. We should kill this bacterial cell. And these antibodies are made up of four peptide chains connected by disulfide bridges or disulfide bonds. 'Di ba? Pag more than two peptide chains, so more than one peptide chain, pwede siyang mag-quaternary structure. So this Y shape here is actually the quaternary structure of the antibody. And the disulfide bonds can be formed by what amino acids? Example, si cysteine. Okay, si cis. These are the different types of amino acids. We have immunoglobulins. We have IgA, IgD, IgE, IgG, and IgM. Here are some of their properties, and this is their structure. The largest is your IgM kasi it's pentameric. It's made up of 5 of these Y shapes together. IgA is dimeric. So, ayan, dalawa siya. Well, D, E, and G are monomeric. Okay. IgA is usually found in mucus, saliva, tears, and breast milk. So, they protect against pathogens. So, yung luha ninyo, yung nakuha ninyo from your mom, which actually protects the baby in the first few months of life, is IgA antibodies. IgD is part of the B cell receptor, which you will see in basophils and mast cells, activates them. IgE is the one that will increase against parasitic worms. So, mag-increase yun siya pag meron kang parasites. And they're also responsible for allergic reactions, so parasites and allergies. IgG is secreted by plasma cells into the blood, and they are the smallest ones. And because they are the smallest, the gamma, immunoglobulin gamma, they can cross the placenta and into the fetus. So, lahat ng IgG na meron si mommy, so yung mga protected siya against, mapapass on kay baby. Kaya important din na si mommy ay fully vaccinated. IgM naman may be attached to the surface of a B cell or secreted into the blood and is responsible for the early stages of immunity. So in initial infection, for example, ang unang tataas are your IgM antibodies. But later on, for more long-term immunity, si IgG na yon. Okay, again, first that you will see will be IgM, but more long-term protection is by IgG. So we are now done with the lecture. Please study for a quiz on these proteins on the week after. So, hindi pa to kasali sa Just Proteins lecture natin for 2F. Okay? Just for 2F, this is not included in the Proteins Part 1 quiz. Okay? So, please read up in your book, supplement lang the knowledge that you have, and have fun. Thank you.